Well, thank you everybody for joining us today. This is our final INN BCNI symposium of the season. We'll pick it up again in the fall. And I'm very delighted to introduce my dear friend and colleague, Petra Ritter, who's agreed to be our final speaker of the session. Petra is currently in Berlin. She did study in Charité and got her degree in neurology, and also did some work at UCLA, UCSD, Mount Sinai, and Harvard School of Medicine in Boston, and toured around the world. One of the innovations that Petra had done early in her career, she was one of the first people to actually do simultaneous fMRI and EEG. And that has helped to sort of support a burgeoning research program that looks at the integration between different modalities and using that to get better information about the brain. Petra is also one of the co-founders of the Virtual Brain, which is how we all sort of got together. And she's driving things quite remarkably with the development of what's called TVB Cloud, which is an amazing achievement, is well supported, and involves a little army of researchers across Europe as well as internationally. And it is an impressive innovation, both in terms of just basic neuroscience, but also the combination of technology. And that's one of the reasons I wanted Petra to come speak with us today, because I think she's really managed to bridge fundamental neuroscience, but use it to actually develop technology that can have direct impact in helping patients. So I think I'll stop there, and then Kelly, does Petra have the capacity to share her screen? Okay, Petra, please, if you'd like to take the stage now, and I'm delighted again to have you with us today, and I'm looking forward to your talk.
Thank you so much, Randy, for this really nice introduction. I'm going to share my screen now. I hope it works well. Which screen do you see, the presentation or the presenter?
The presentation.
Wonderful. So it's a lucky day. Thank you very much for having me here. It's a great pleasure to be able to present the Virtual Brain Cloud, enabling complex simulation with multi-level health data. And I assume that most of you in the audience have a background in neuroscience, and probably we all are driven together by the question, how does the brain work and how does it not work in disease? And we all know that the brain is a complex system, and you can make many different kinds of observations at different spatial scales and temporal scales. And the challenge is to bring all the different snippets of observations together in a consistent theory of the brain. And one step towards this is the Virtual Brain Simulation Platform, or more in general, the brain network modeling approach that Randy has already mentioned. There we construct biophysically grounded network models, mathematical models of the brain that simulate the activity that we can measure in real life. So for example, firing rates, local field potentials, EEG, MEG, fMRI signals, this all can be simulated by an underlying biophysical model that is driven by the structural connectome. And this can be done for humans and also for animals. Here you see a macaque virtual brain in the right lower corner. So the software, of course, I think everyone knows here in this audience. This is the Virtual Brain landing page. And actually we got now 10 years old. It was published for the first time, or released as open source software in 2012, if I'm not wrong. And we are approaching, as you can see here, the 40,000 downloads. I just 1 hour ago took a screenshot, so it should be still about this number. And you see it's still attractive. So just in the past 30 days, there were almost 650 downloads. And you also see the latest publication here, Brain Simulation as a Cloud Service, the Virtual Brain on eBrains, This is one of the topics that I would like to present and to cover today. And yeah, needless to say that Randy and Victor in Marseille and Jochen Meersmann in Stuttgart and myself, we are leading this initiative together. So one of the earlier works that we published now a couple of years ago is the demonstration of the capability of such brain network modeling approaches in general in bridging the scales between different entities and observations. So what you see here on the left side is a simulation of about 20 minutes of resting state activity. And below you see how for a single node that has been picked from this brain network model, we infer the excitatory and inhibitory population firing that we could not measure non-invasively in this person. So by reproducing the resting state dynamics, we are able to capture at the same time the firing rates of the excitatory and inhibitory populations. And here in this diagram, you see in the upper panel the simulation results analyzed with different methods across different time scales. From very fast scales, here we have a time window of about 100 milliseconds on the left side, to very slow scales of several minutes, specifically 22 minutes on the right side. And on the lower panel, you see the experimental data corresponding to these simulated signals. So for example, here we see the relationship between firing rates and the phase of the local field potential and the alpha band. And below you see the corresponding well-established relationship that has been acquired or measured in monkeys. And if we go to the next panel, B, we see the relationship between the amplitudes of inhibitory postsynaptic currents and excitatory postsynaptic currents. And below you see the same measurements in real life in rats and so forth. So here you see in C the relationship of firing rates and the alpha band power. And below you see the measurements in monkeys. And panel D shows the relationship of EG, alpha band power, and the fMRI bolt signal. Panel E shows the scale invariance and resting state data. And the last panel, which I don't see because I see the heads, I have to change that. We see the functional connectivity dynamics in our simulated data and the corresponding empirical data. So with a single model, and in this case we had actually 15 brain network models of 15 individual healthy adults, we can link these different snippets of observations in a single consistent model, and we can relate the processes mutually. So we really get a mechanistic causal understanding how the different pieces of observation are linked together. And this is what we want to achieve. We really want to get a biophysical understanding, a causal mechanistic understanding of the processes in the brain, because if we understand them, we are able to manipulate them and to push them, for example, back in the right direction in brain disease. This is another example where we really obviously need brain network modeling to understand the complex evolving dynamics. So what you see here on the left side are traveling waves. Traveling waves can be measured with EEG, with MEG, or here in this case it was intracranial EEG. And they have been related to many different types of cognitive function. And the special attractiveness of these traveling waves that can change their characteristics, they can change the direction, they can change the speed, and they can bind together distant parts of the brain. They have a wave front that moves across the brain, and by this they synchronize distant parts of the brain. And with this, they are candidates for a mechanism that channels the information flow in the brain and also leads to synchronous spiking in distant parts of the brain that would enable to consolidate, for example, memories in distant parts of the brain. And in order to understand the mechanisms that underlie the generation and the change of the characteristics of those traveling waves, we need complex models. This is a work of an ongoing project of Dominic Koller from my group. He is doing his PhD on traveling waves. We have here a very dense network model, and you see that by actually changing the excitation inhibition ratios in the different parts of the brain, we can manipulate the direction, also the speed of these traveling waves. And this type of investigation would not be possible without a brain network model. So in order to understand really the generative mechanisms of such complex dynamics, we need a complex system. This is another example where we show that changing a single parameter of such a brain network model, in this case, this is a global scaling parameter, we can, in a certain window, in a certain range of parameter values, reproduce different features that we observe in empirical data. For example, functional connectivity and functional connectivity dynamics. Here you see the empirical data, here you see the simulated data, and also certain features that we see in the EEG, which you see here in the middle. So now we have reached that window, and suddenly all the biological plausible structures emerge in our simulated outputs. So I think our team in Berlin, but of course many other teams, including the one led by Randy McIntosh, we have demonstrated that many kinds of dynamics can be generated by brain network modeling, and certain principles can be derived with this approach. But what really is driving our curiosity is how are the dynamics now linked to cognitive performance and behavioral performance. And in order to investigate this further, we have used the data of 650 individuals of the Human Connectome project. And we choose those data sets that contain the functional MRI data, structural MRI data, and also cognitive scores. And we have reconstructed 650 virtual brains of these individuals. Yeah. And the goal was to relate a measure, fluid intelligence, that is actually a measure of deep thinking and problem solving, to the dynamics, and to understand how the differences between the 650 individuals and performance and fluid intelligence tasks emerge. This is very new data. It's not yet published, so probably you hear it for the first time, and I'm very happy to present this here today. So how do we measure fluid intelligence? Fluid intelligence in the Human Connectome project has been measured with the Penn Progressive Matrices test, PMAT. Here you see one example of one of the 24 tasks. So the subjects are asked to identify the most appropriate match among the given options, and they have to identify a hidden rule that underlies this match. Now I ask you, what would be the right response here? Who knows that? Who dares? So actually, that there's some silence. I hope this doesn't mean that I'm disconnected. I assume that you are still there, but it means it needs some thinking. Yeah, even though this is one of the easier tasks, so the difficulty is increasing with each task along the 24 tasks. It needs some thinking. Yeah, you have to develop a hierarchy of decisions and keep some information in your working memory, and it takes some time to come up with a solution. In this case, would be the item 8. Yeah, so if we look in the first row, we see the two pieces move to one step to the right and the left, and then one further step to the right and the left, or counter clock and with a clock. And here happens the same. The two pieces move to the right and the left, one step further right and left, and so forth. And here, of course, you can apply the same scheme, and then you would end up with this pattern. So this is a type of thinking that is tested with the PMAT test. In contrast, for quantifying the processing speed, the NIH toolbox has the so-called dimensional change card sort test, or card sort test. And in this type of test, the decisions can be made very easily and rapidly. So they are basically no-brainer tasks. And here in this example, the subject is initially asked to sort the cards by a single feature, that could be the color. And then subsequently, the subject is asked to alter the strategy and to sort the card according to another feature that could be the shape. So in the first case, one would map red to red, or black to black, gray to gray, and here one would map the rabbit to the rabbit. And in this example, it is very clear that it doesn't need a lot of thinking. Yeah, it is a simple feature that can be recognized, and it doesn't need to be kept in the working memory for a longer time period. And since the strategy also changes over time, it is good if actually you forget the history and overwrite the content of your working memory in the next task. So now if you have thought that intelligence is related to faster thinking in general, these results that come from the Human Connectome Project data will actually show you that this holds not true. What we see here, again, are the 24 questions of the fluid intelligence task, the PMAT task. And on the y-axis, we see the degree of correlation with, between the reaction time and either the fluid intelligence, the G factor, shown in bright blue, or the PMAT test, the fluid intelligence test, shown in dark blue. And here we have group averages on this side. Here you see the individual subject results. And what we see here is that up to question 8, we have actually negative correlations, but for the more difficult questions of the fluid intelligence tasks, we have consistently positive relations between intelligence and the reaction time. What does it mean? This means that the people who were quantified as more intelligent, either in the G factor or in the fluid intelligence task, they took longer time to take a decision, and they came up then with better decisions. So now the question is, how would the fluid intelligence or the general intelligence relate to the dynamics of the brain? And this has been explored before. This is nothing new, but we did it again with the Human Connectome Project data. And we found, as others before, that there's a very low correlation, but some correlation between the functional connectivity and the data, resting state data. This was 1 hour of resting state, fMRI data, and the general intelligence, and also some degree of correlation between the functional connectivity and the fluid intelligence. But it was at the border of significance. It was not really strongly significant. If in contrast, we looked at the relationship between functional connectivity and the reaction time and this fluid intelligence task that needs a lot of thinking, we found a very strong correlation. Yeah. So those subjects who had higher or longer reaction times, who took more time to respond in this task and also performed better, they had more synchrony in their functional connectivity. The activity in their brain was higher correlated. So now we had the opportunity to construct the brain network models of these 650 individuals. And we did it in a classic way. So we had 379 nodes, and each node was equipped with such a population model consisting of an excitatory and inhibitory population. And they were mutually affecting each other with long-range connectivity that was simulated via the structural connectome that we derived from the diffusion tensor imaging data and tractography. But as a novelty, we not just included the excitatory long-range connectivity, but also feed-forward inhibition, long-range inhibition, which is a common phenomenon in the brain and which increases the biological realism of our brain network model. So the new connections are shown here. There's not just going excitatory drive to the neighboring and distant excitatory populations, but there goes also excitation to the inhibitory populations, which leads to feed-forward inhibition. And this is a common principle that is seen in the brain. So with this, we can now tune the ratio between excitation and inhibition, the ratio between long-range excitation and feed-forward inhibition. And this improved considerably the achieved fit between empirical functional connectivity matrices and the simulated functional connectivity matrices. So here you see what is happening. If we tune the relation between long-range excitation and feed-forward inhibition, which I show you here in the little video, we can change actually the correlation between the signals, the functional connectivity. So here you see the ratio of excitation and inhibition. Here you see the functional connectivity, and here you see the signals, and you see the correlation coefficient. This just illustrates the populations and their connections, and you see that they're getting, here the feed-forward inhibition is getting fatter, so the ratio, I ratio is getting smaller, and now vice versa. So with this simple trick or mechanism, it is possible to tune the degree of the functional connectivity in the brain. And we developed a tuning algorithm, and you see here how this evolves over time that fits the simulated functional connectivity to the empirical functional connectivity by tuning the ratios of feed-forward inhibition and long-range excitation. And with this, we achieved very good reconstructions of the individual functional connectivity matrices. So now we had models that simulated the functional connectivity that we could also observe in the empirical data. And we had, of course, the underlying state variables. And we're now able to relate the characteristics of these underlying state variables to the performance of the individuals behind the virtual brains. And this is what we did here. We looked at the simulated synaptic amplitude and synchrony. So here you see the degree of correlation of the input currents that drive the populations, the nodes in the brain. And here you see the amplitudes. And on the x-axis, you see the reaction time in the fluid intelligence task. And we see very strong correlation between the input currents of our model and the reaction times of the true empirical reaction times of our subjects. Yeah? So those who were slow had a very high correlation in their model of the input currents that drive the nodes. And also those who were slow, they had pretty low amplitudes, lower amplitudes than those that reacted fast, of input currents that drove on average their nodes in the brain. So there was a significant relation between the simulated synaptic activity variables that we couldn't measure in the empirical data, but we are reconstructing them with our models, and the performance, the reaction time. So now the question was, how do these features from our simulations relate to the empirical functional connectivity. And also there we found a strong relationship. As you can see here, higher input currents in our model were associated with lower empirical functional connectivity. And higher correlations of or synchrony of our synaptic input currents in the model were related with higher empirical functional connectivity. So this suggests that when we tune the functional connectivity in our model, we at the same time also tune the input currents, the amplitude of the input currents that drive the nodes, and the degree of correlation of the input currents that drive our nodes. And this indeed is the case. This is illustrated here with this little video. Here we see again the ratio of excitation inhibition, and we can change that. And at the same time, we change the degree of functional connectivity. Yeah, so with high excitation inhibition ratios, we have a high positive functional connectivity. With low excitation inhibition ratios, we have an anti-correlation. And with intermediate ratios in a balanced state, so to say, we have asynchrony, there's no correlation. And at the same time, this also changes the input currents that drive our nodes in parallel, but it also, so it changes the correlation, but it also changes the amplitude, as you can see here. And the highest amplitude we see at a balanced state, at an intermediate ratio of excitation inhibition, where we also have asynchrony and no anti-correlation and no positive correlation in the input currents that drive our nodes. So now we have a brain network model that is capable of simulating the dynamics, the functional connectivity that we see in the empirical data. And we have seen that some features of this model relate to features, cognitive features, but also dynamics, functional connectivity in the empirical data of the brains of the subjects. So now the question is, how can we understand how the dynamics in our brain network model that change with the performance of the real subjects influence the capability for performance? In order to investigate this, we employed a frontoparietal circuit model that is capable of decision-making and working memory. And this is a pretty old model. For the first time, this type of model has been published in 1997 by Schmidt and Brunel. And they simulated working memory with this model. So basically, it's two populations. They are mutually inhibiting. They have very strong self-excitation. And with this, they can reach or switch between a low attractor state and a high attractor state. And in 2006, for the first time, Wong and Wang used this type of model to simulate decision-making. Yeah, because of the mutual inhibition, there is a winner-take-it-all mechanism implemented. So the two populations are competing, and therefore a binary decision can be taken between the two populations. And this model has been further advanced by Murray, Jaramillo, and Wang. And this was published in 2017. And we used and implemented this frontoparietal model in our 650 virtual brains. So now if we look at the performance of this frontoparietal decision-making and working memory model, we see that the performance differs independent of the input amplitude and the input current correlation. So here in the upper panel, you see the dependence of the input amplitude to the posterior parietal cortex and the prefrontal cortex. Here color-coded, you see the correct decisions of the circuit model. And here the integration time it takes to achieve an attractor state or a decision. And in the lower panel, you see the same, but for the degree of correlation of the input currents to the posterioparietal cortex and the prefrontal cortex. And we see that by changing the input amplitude or the degree of correlation, the percent of correct decisions can be altered, and also the integration time changes. So there's a dependence, obviously. Same holds true for the robustness of the working memory states that can be elicited. So what we see here are three scenarios with three different input current amplitudes, low, middle, high. And the diagrams show the stimulus that elicits, the stimulation strength that elicits the working memory trace, versus on the x-axis, the strength of the recurrent structure, so the recurrent excitation strength. And these are bifurcation diagrams. The red areas show the areas where we have 100% robust working memory trace. The gray areas show the region where the stimulus would not elicit a working memory trace, it's too weak. And the blue areas show the regions where a distracting stimulus would lead to a disturbance and deletion of the working memory trace. So the red areas have very stable working memory that cannot be deleted. The blue areas have unstable memory. And you see that depending on the input current, this bifurcation diagram changes. Yeah? The properties of the working memory capacity change independent of the input current strengths. And this is also illustrated here. Here, the three different input current strengths are indicated by these three colors, blue, red, and green. And we see here the override threshold. So for the lowest input current, we have the highest threshold. That means that the trace, the tractor state, is most stable, and it takes the biggest effort to override this trace compared to the situation when you have higher input currents. Here on the right side, you see the memory induction threshold. And again, for the lowest input current, you have the highest threshold. So it takes a very strong stimulus to elicit a working memory state. Yeah? So if you have just noise or small stimuli, that would not lead to a working memory state. But if you have a strong stimulus, you would go in a tractor state. And at the same time, you have a high override threshold, so the attractor state would be more stable. So with a low input current, you have more stable attractor states that need more effort to be activated. With higher amplitudes, you have lower thresholds, so already maybe random stimuli would be sufficient to trigger a memory state. But the memory state is also less robust and can be easily overwritten. So now, of course, the interesting question is if we plug such a circuit model that is capable of generating working memory, attractor states, and taking decisions in our 650 virtual brains, do they perform differently, and does the performance relate to what has been measured in the true subjects? So what we see here is the decision-making performance of the model on the y-axis. And on the x-axis, we see the fluid intelligence score measured with a fluid intelligence task. And we see a very strong correlation between the performance of the circuit model, the generic circuit model in our virtual brains, and the fluid intelligence as measured in the people behind these virtual brains. If we look at the right side, we see that the time that it takes to come to a decision is also correlated with the fluid intelligence score of the individuals. So the time it takes in the model for the circuit decision-making and working memory circuit to move to an attractor state takes longer for individuals with higher fluid intelligence in real life. So now we have the model, the brain network model, that is capable of reproducing performance features like percentage of correct decisions, and also the time it takes until the decision is reached. And the model provides us with also state variables that we now can analyze to identify the mechanism that leads to such different performances. What we see here are 5 scenarios for 5 different input currents again. And we see the firing rates of a population of such a circuit model consisting the prefrontal postparietal circuit model consisting of the 4 nodes that we have seen before, the 2 competing nodes A and B. One is coding the distractor, and one is coding the correct decision. In our case, this is A. And we see the firing rates, how they build up over time. And with the naked eye, and on the first side, we see that there are different temporal dynamics for the different input current strengths. For high input current strengths, the firing rates immediately jump to a high attractor state. For the low input strengths, it takes quite a while until the low attractor state is reached. And the little arrows here, they indicate interesting time points in terms of the prefrontal node. Yeah. So they mark low attractor state of the prefrontal node, then the ramping up of activity in the prefrontal node, and the high attractor state in each of these scenarios. And here you see the time it takes to come to a decision depending on the input current. So now we have the opportunity to look at these phase portraits. And again, we have the same configuration. We have the situation for the 5 different input strengths or amplitudes. And the rows indicate the 3 different time points that were marked with the arrows that capture different states of the activity in the prefrontal cortex. So the low attractor state, the ramping up phase in the middle, and then the high attractor state in the prefrontal cortex. The axis here of these phase portraits span the activity in the posteroparietal cortex of node A and B, yeah, the 2 competing nodes. And the color codes the magnitude of the flow fields. The arrows show the direction. The green and the blue line show the null lines. The brown line shows the separatrix. We also have 1,000 simulations, simulated trajectories projected on top of this phase portrait. They are indicated in green and red. Depending on in which decision they end up, green would end up in the attractor state in the lower right corner, indicated with a white circle or a black ball. And the red marked end up with the competing decision, the competing population and its attractor state in the upper left corner. So I mentioned already the balls, the black balls are attractor states, and the white ball is a saddle point. And now if we focus in the beginning on the middle column, we see that the phase portrait is quite symmetric. So we have 2 attractor states. We have here on the diagonal a saddle point. And we see the flow fields for the trajectories. And so you see that the trajectories would relatively quickly have to move to either one of the 2 attractor states and either end up in the correct decision for population A or in the wrong decision for population B. So now in the moment, the second line here in the middle, when the frontal activity in the circuit is ramping up, we see that the configuration of the phase portrait changes. The separatrix moves a little bit more to the left upper corner, and the pull towards the attractor and also the basin of attraction towards the attractor in the right lower corner is getting stronger. And in this example here, and also in the previous slide, that there would be a pull to the right decision. Yeah? The trajectory, as you can see here at the bottom, ends up in the correct attractor state. So now if we look at the scenario where we have a lower input current, we see that there's a reconfiguration. The saddle point that was in the previous case here in the upper right corner now moved to the lower left corner. And this changes completely the pattern of flow fields. So what we see now is that initially the trajectories are kept through the saddle point here in the lower left corner on the diagonal. Yeah? So there's no push to either of the two attractor states, but they are kept more on the diagonal, which gives them more time to integrate evidence and to average out noise. So now if we look in the second panel, this is when the prefrontal activity is ramping up. We see that the saddle point moves, yeah? And this puts the trajectories in the position to leave the diagonal and to move to one or the other attractor states. And it's becoming even more obvious what is happening here in this scenario where we have the lowest input current strength, and it takes the longest time to come to a decision as indicated here on top, and we have the best decision-making performance, also the highest ratio of correct decisions. Here we see that because there's a higher input, synaptic input current strength, at the beginning we have a reconfiguration of this phase portrait. We have the saddle point, we have two saddle points actually. So they have split in two parts, and there's an additional attractor that emerged here in the middle on the diagonal. And this now keeps the activity of the competing population A and B at the beginning with this middle attractor space. So only if the prefrontal cortex then starts to ramp up, we see that the third attractor is resolving, it is disappearing. Also the split up saddle nodes are merging now to a single saddle node. And this puts the system in a situation when the trajectories are released and can either move to the right bottom or left top attractor. So it takes the input from the prefrontal cortex here to move the configuration from this state to that state and to release a decision. So now if we move to the other end of scenarios with very high input strengths, and as you can see here, the lowest decision-making performance and the shortest time to come to a decision. We see that here we have another scenario where we have again two saddle points that are split up and an attractor state in the middle that pulls the trajectories on the diagonal in a high attractor state. Yeah, so population A and population B from the very beginning are put in a very high state. And it takes only a very small additional kick to move the trajectory then to either of the two attractor states. So it could be a little piece of noise that is sufficient to come to a decision. There's not much time for evidence accumulation. But on the other hand, you could consider these high energy states of the two populations as a state of attention or alertedness. And I think Gustavo Deco once came up with the metaphor of the tennis player that moves from left to right and right to left to stay in such a high alertness state, high activity state, to more effectively react to an incoming stimulus. And this is exactly the scenario here. So the activity in both populations is high. It takes only very little additional activity to come to either of the two decisions, can be wrong or right, but it takes not long time. But accordingly, the performance is worse than if there's more evidence accumulation on the other end of the scenarios. So this shows how we can actually use the tools that are provided by dynamical systems theory, mathematical tools, to understand what is happening in a complex system that leads to differential behavior and different scenarios. And this is also illustrated here a little bit more dynamically. Here, it might be a little bit confusing. Just follow my mouse cursor. We see the changing input amplitude. This is also shown here. So we move this red dot, and by changing the amplitude now to a lower state, we reconfigure the phase space. And according to the final input current strengths, we see the performance will vary. So in this case of an intermediate input current strengths, we have a performance of 81.7 correct decisions and an integration time of 0.81 seconds. And here you see how the trajectories are evolving in the phase space. And here you see actually the time series of the four competing populations. And if we now move the input current, we now move it to a higher state, we have less correct decisions, and we have a smaller, shorter integration time. And we can do this, of course, in the other direction. Now we lower the input current, we reconfigure the bifurcation, the phase portrait, and we get now a higher rate of correct decisions and have a longer integration time. So now, of course, we are also interested not just in the input strengths, but also in the degree of correlation of the synaptic input currents, because we have seen earlier that they are also related to the performance. And this means we have to look at the phase portrait of a stochastic system. Yeah? So there's not a single gradient field as in the previous slides, but there are many different scenarios and probabilities, and therefore now we have to work with glyphs. And the glyphs indicate the entropy of the gradient fields. So here you see the color scale, and the shape also indicates the entropy. If we have a round circle, like here, for example, the gradient fields, or the magnitude of the gradient fields, is moving or pointing in all directions to a similar degree. If we have these stretched glyphs, then the gradient fields are more pointing in one direction. And we see that for higher degrees of correlations, we have more stretched gradient fields, and for lower degrees of correlation, we have more round gradient field glyphs, indicating that in this scenario on the right side, the trajectories are more pulled towards the diagonal, while in this scenario with lower correlations in the system, the pull goes in all directions, including the directions of the two attractor states. So the trajectories on average are not held on the diagonal, but moving faster to one of the two states, which leads again to a shorter evidence integration time and to a worse decision performance. And if we now average the speed, the flow field speed according to the degree of correlation of the input currents, we see this U-shaped form. It means that with an intermediate correlation, we have the lowest speed that allows the longest evidence integration time. And this is here dynamically also illustrated. Just focus on this part of the slide. And we move now the red dot, not for the input current amplitude as before, but for the degree of correlations and synaptic input current correlation. And this reconfigures systematically the shape of the glyphs that are representing the stochastic flow fields. And with this also change the trajectories that we would see in real simulations. So taken together, I hope I could show you in a more or less understandable way that the brain network models enable us to not only look at the dynamics, which are already interesting on their own, but also relate the actual dynamics to cognitive performance. And here in this study, we have shown that through the tuning of the ratio of long-range excitation and feed-forward inhibition, we can manipulate the degree of synchrony in the system, the functional correlation, the functional connectivity, that the excitation inhibition ratios also modulate the synaptic input currents that drive the nodes in our models, and the degree of correlation of the synaptic input currents, and that the strengths of these input currents and the degree of correlation directly influence the decision-making performance and the time until decision in our decision-making working memory circuit. And with this, I end this study, and I now completely switch gears. I wanted to now on a little bit more superficial scale. I just look at the time and I see it's pretty late, took longer than expected. What else we can do with the virtual brain and what we have developed on top of the simulation framework that exists now since 2012. To make it accessible and usable for the research communities. So needless to say that, of course, brain network simulations is not only interesting to understand the fundamental principles of brain function, but also to understand diseases, and many studies are ongoing there in epilepsy, brain tumors, psychiatry, stroke, dementia, traumatic brain injury. And there are three principal directions how brain network modeling can be utilized in the context of diseases. One direction is, of course, to understand the mechanisms, and this is a highly relevant direction. But another direction is to use the simulated features that we cannot measure to enrich the feature space, hoping that the features that we derive from our models are closer to the actual mechanisms and the neuronal interactions that are related to disease, and improving the classification of disease subgroups, for example, or the prediction of a disease trajectory. And the third pathway is to use the virtual brain to do in silico planning of virtual therapies. Here, this is a study that I want to mention shortly, where we included as another modality positron emission tomography data in our models. So in this case, this was ADNI data of patients with Alzheimer's disease, healthy controls, and mild cognitive impairment. And we had the PET data that provided information about the load with the pathologically accumulated protein beta amyloid in Alzheimer's disease. And we had this distribution not only for the Alzheimer's disease patients, but for the healthy controls and the mild cognitive impairment patients. And from the literature, we know that in the vicinity of this beta amyloid, the function of inhibitory interneurons is impaired. And this is a mechanism that we implemented in brain network models. So the degree of burden of beta amyloid in each individual region had a direct effect via such a transfer function on the degree of coupling between the inhibitory neurons and the excitatory cells. And by implementing this load of beta amyloid via this coupling function, we got a slowing only for the Alzheimer's disease patients and the simulated EEG. We didn't change anything else. We only included the PET data in the models and got a slowing exclusively for the Alzheimer's disease patients. And this was significant. And I jump over these. These are very nice mechanisms that show how exactly this can be traced backwards, so how this protein changes the frequency of the oscillations by changing again the phase portrait and then leading to slower oscillations. But I want to also mention this study that just got published, where we actually provided for the first time, and this was Randy also, evidence that or actually, but probably I shouldn't say for the first time, because there was an earlier study with Joel and also Randy that pointed in the same direction, but this is additional evidence that using the features from our simulations, we can improve classification of disease groups. This is what you see here. We see that when we combine simulated features and empirical features, we significantly have a better classification accuracy based on the data that we have between Alzheimer's disease, mild cognitive impairment, and healthy control groups. And interestingly, we can also identify the features that most significantly contribute to this better classification. And this is the alpha band local field potential activity, in particular in the thalamus, but also in the occipital cortex and dorsal stream, basically in the areas where we find the alpha rhythm and also the alpha rhythm slowing that we don't have in the ADNI data, but that we inferred through our simulations. And it is possible now to use these brain models for intervention testing. Here, in this case of our Alzheimer's disease patients, we tested the effect of memantine, an NMDA antagonist that is given also in Alzheimer's dementia for temporary relief of cognitive symptoms. And it's an NMDA antagonist. And this mechanism has been implemented here by changing independence of the drug dose, the coupling between excitatory interneurons and pyramidal cells. If we give this virtual drug, you see a normalization of the slowing that we find in the simulations of the Alzheimer's disease patients to the normal baseline. But the beauty is that we now can look again after a mechanism that leads to the protection or the improvement, the cognitive improvement of the subject. And this is what you see here. So we see here the input that drives the pyramidal cells. Here we see the dose of the drug, the NMDA antagonist, and here the output of the pyramidal cells. And the problem, and one of the theory candidate mechanisms leading to dysfunction and dementia is the hyperexcitation. So due to the loss of function of inhibitory interneurons, there's an hyper excitation of the pyramidal cells, which leads to excitotoxicity and supposedly also cell death. Now, if we increase the activity that drives the pyramidal neurons, automatically also the output of the pyramidal cells increases. But if we also increase the dose of the NMDA antagonist, we see that we build in such a little buffer that compensates and keeps a zone where we do not see an increase of activity in the pyramidal zones, pyramidal neurons. And this could be the protective effect of NMDA antagonists in Alzheimer's disease. So now I come really with big steps towards the end of my presentation. I think I forgot to ask how long it shall be, so I didn't get any constraints. I can talk as long as I wish.
You can finish it on, that's fine.
So what you see here is the landing page of eBrains. eBrains is the European infrastructure of digital infrastructure that has been built by the Human Brain Project. And I think the number that you see here is from March. So I was inquiring about how many user accounts, and at that time it was 5,343. Probably it changed a little bit. But this infrastructure has the goal, and has also achieved part of that goal already, to provide a single entry point for digital neuroscience research for the research community, not only in Europe, but globally. And there are different categories of services: data and knowledge services, atlases, simulation services, brain-inspired technologies, for example, neurorobotics platforms, and medical data analytics tools. So this is an overview of the user community, just to give you an idea. So most of the users presently come from Europe, and in Europe, most of the users come from Germany, followed by Italy. But a little bit less than a quarter of the users also come from other parts of the world. For example, from North America or from Asia. From Canada, we have 3% now. Not so bad. But yeah, so of course the infrastructure is open also for North America and for Canada, and we are very happy to increase the percentage of users also outside Europe. One effort that we undertook over the past years is to make the simulation services also available via this infrastructure. And this is a complex scheme, and what it should illustrate is just that the simulation services which you see here under this virtual brain cloud sign, the simulation engine, but also the processing pipeline in a containerized form, another framework for multi-scale co-simulation, which I also quickly will show you in the next slides, and some other tools for personalized brain simulations. They have been integrated with the core functionalities of eBrains, which are providing collaborative workspaces. So there are in the cloud possibilities to upload your data, to create sandboxes, invite your collaborators, or stay completely private and work with your data or grant access to your data. There's, as I mentioned already, the possibility for cloud services. So you can provide tools in the cloud. The virtual brain can be directly started in the cloud. So you find that the service actually, if you go to this website, and there's a high performance computing backend powered by 5 supercomputing centers in Europe. And the services, for example, the virtual brain service also utilizes this HPC backend for compute costly simulations. The same is true for the processing pipelines. And these tools are interfaced with RESTful APIs mostly, but they're also linked with digital atlases, for example, that are also available in eBrains, or with a neurorobotics platform. And in Europe, data protection is a big topic. There's a general data protection regulation, so everything is encrypted and sandboxed, and there's access control mechanisms to make it compliant with the data protection requirements, and to keep the data safe, which are, as you all know, highly personal data, because these are actually fingerprint data. Even if the names and birth dates and stuff is removed, they are highly individual, and in principle it is possible to re-identify an individual based on their brain data. The nice part is that it's possible now, and we are developing several tools that go in that direction that have not yet published. So I will also not go in the details, but to map knowledge from other repositories, not just digital atlases, but also knowledge bases, pathways, inventories, to link the signaling cascades that have been derived through, for example, literature mining algorithms going over more than 30 million of publications in PubMed. And we have developed a tool to map these cascades to the 3D space of the brain in order to then link them via such functions, similar as we linked the PET data, transfer functions, and the PET data to the function of the inhibitory interneurons. We can link such signaling cascades that are changed and associated with diseases to certain parameters of the brain. And of course, also Atlas data like receptor densities. This is an example where the Zebra Python client, we can access distributions of receptors from digital atlases and e-brains, and everyone can do. So this is again open for everyone. And we can lay these distributions of receptors on top of our virtual brain and change accordingly the parameterization of our local models. So we can take into account biological heterogeneity in an informed way. Another very recent development, which I find extremely exciting, is an interface that also has now been established with a neurorobotics simulation platform. So there's not just the virtual brain simulator, but also the robotics simulator. And here we connected the virtual brain with these two hands, and we reproduce a phenomenon with the simulation in the robotic hands that has been extensively investigated and published. A certain behavior that can be observed when doing this antiphasic movement task. At a certain higher speed, the fingers will move in phase, so that you cannot keep up the antiphase behavior. And the switching dynamics can be nicely captured by the dynamics of the virtual brain and translated in a visible output with a neurorobotics platform. And then the also late development, which I find extremely promising and exciting, is a multi-scale co-simulation where one can define some of the nodes at higher detail. This is achieved through co-simulation with different simulators. We now have an interface with Nest and with Anarchy, which is also spiking neural network simulator. And we are just now collaborating with the NetPine people to also build the possibility to do co-simulations with a neuron simulation framework. This is also available already on eBrains on the website that I showed before and can be directly utilized. So if you're interested in setting up such a co-simulation, it is possible now to define some of the nodes at finer scale of spiking network neurons and to run the simulation of the large scale network and the finer network in parallel. And this has been actually exploited for deep brain stimulation. We are very happy that now this proof of principle just also was accepted at the European Journal of Neuroscience, obviously. So I think, yeah, I always think it is experimental neurology. I think the references were wrong in the previous slides and I put the wrong journal name. But what we do is we define the basal ganglia here at spiking network detail and the rest of the brain with the typical mean field approximations and population models. And this is how a simulation then looks like with different stimulation paradigms. This is the spiking trains and the basal ganglia and the thalamus. This is the cortical activations. And here you see also the firing rates in the off stimulation state and the on stimulation states with 3 different stimulation types in the thalamus. The beauty is that this is what you see here, and I think here this is the wrong journal name, that for the 3 different stimulation types, we get the same firing rates in the thalamus, which is relevant for treatment in Parkinson's disease because the activity is probably one of the pathologically changed activities that need to get counteracted to get an improvement in the motor behavior in Parkinson's patients. But what we also see is that even though at the thalamic level, the impact of the deep brain stimulation and different nuclei was the same, had the same effect, At the cortical level, it was quite different. If you have a monophasic stimulation, we get a deactivation in the motor cortex, and if you have a biphasic stimulation of the subthalamic nucleus, we get a strong activation. And this is what we get if we stimulate the globus pallidus and ternus. These are the structures that typically are stimulated in deep brain stimulation. So this is just proof of principle. The hope is that on an individual subject level, we can refine the approach of multi-scale co-stimulation and really establish this as a tool to improve the optimization of the stimulus for the individual patient. I think this is the last slide. This is a team in Berlin. I would like to thank everyone there. They're all contributing to the different projects that I have shown. And of course, the other collaborative partners on the big projects. And first of all, the partners on the virtual brain. And they are foremost Randy, my long-term friend.